Before we get started, I have a quick favor. I've been self-funding the Finding Genius podcast for 5 years now. I've done over 3,000 episodes. And as you can see on YouTube, we're up over 1 million views on the channel, which is fantastic. The next thing I really want to push on is to get up to 10,000 subscribers. Because once we do, we'll be able to put a donate button and we'll be able to solicit donations to help keep the podcast running and to also get the Finding Genius Foundation moving along. We have a big project studying anxiety, depression, and PTSD and working on a product to help people overcome these problems because I've seen them explode recently after the last 2 years of the whole virus situation. So if you would, please subscribe to the podcast. That would help us tremendously. Give us a thumbs up and check in the description for Buy Me a Coffee. It's about $5. If you could buy me a coffee, I'd really appreciate it. It would help keep the channel going, and I love coffee. Thank you.
Forget frequently asked questions,
common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs, the Finding Genius podcast host. Today, my guest is Stephen Maher. He's a professor in translational oncology. We're going to talk about the role of microRNA and random mitochondrial DNA mutations in cancer. Professor Maher graduated from RCSI with a PhD in oncology, and then in 2004, he got a fellowship from the National Cancer Institute in Maryland. Spent 3 years working on novel immunotherapeutics for cancer. 2007, went back to Ireland and Department of Surgery at Trinity College Dublin, St. James Hospital as a research fellow, and on and on. Got a lot of experience in cancer and looking forward to speaking to him today. So welcome, Stephen. Thanks.
Thanks, Richard. Happy to be here.
Yeah, what got you interested in cancer, 20 some odd years ago, and why is the interest continued?
Yeah, I've been interested in cancer for a very long time. I think when I was an undergraduate student, it was a disease that even as a young person, most people had heard of it when you hadn't heard of other things. So it was kind of a word that everybody'd heard about, but you didn't really know that much about it. And then as I went through university, I learned more about it, and it became quite an interesting, conundrum in that a lot of people get it. Not an awful lot of people know much about it, and it can be either very hard to cure or very easy to cure. So I suppose, 20 odd years ago, I decided when I was doing a PhD that I wanted to focus on oncology, and throughout the years I've largely focused on understanding pre-malignant conditions, so why patients develop cancer from underlying kind of conditions that would increase their risk, and then largely in treatment and all the different treatment options. So how we treat it, why it works and why it doesn't work.
What's the percentage, I know this is hard to say, but is there any data that says X% of people that get cancer kind of knew they were going to get it based on what they were doing versus it was a total surprise?
Yeah, I mean, cancer is something. So, a lot of people will say, well, why do we get cancer? Why do so many people get cancer? And, historically, we have known that, I suppose historically, it was very difficult to determine what the cause was of cancer. For the longest time, it was thought that, there must be one particular thing that causes cancer, like if it was contagious or was caused, through occupational hazards or that. We know that there's lots and lots of things that can cause cancer. Just by being alive. I mean, if you lived in a bubble and never got exposed to anything, kind of bad, just by virtue of the fact that we are organisms that have renewable tissues that need cells to divide and grow and multiply, just that very fact makes us susceptible to the development of cancer. And then, on top of that, we have occupational risks. We have things like infectious agents that can increase our risk of developing cancer kind of at an earlier age or at some point. So the biggest risk factors for the development of cancer, are aging. And so living longer puts you at increased risk, and being a multicellular organism. So yeah, you will have people who, will kind of have a hereditary element to it. About 5% of cancers have have a hereditary aspect to it where you can link it in within families to particular types of cancer. But the vast majority of cancer, kind of occurs randomly outside of, having a job that exposes you to, say, chemicals or that, that are known to be carcinogenic.
Well, maybe not randomly, but we don't know the cause,
Yeah. So, well, that's the thing. We don't know the exact cause of cancer. We do know that about 40% of cancers have some sort of modifiable risk factor, whether, it's environmental or occupational or, infectious agents driven. So, about 60% find it very difficult to narrow down exactly why somebody has developed it.
And what is your current research focused on? The bio mentioned microRNAs. So what are we looking at?
Yeah, so we have spent, I suppose, 20 years nearly. I've spent 20 years nearly, kind of studying cancer treatments. I'd started off with immune kind of based therapies. My PhD looked at interleukin-2. Interleukin-2 was kind of the interleukins were one of the first, what we call cytokines, ever discovered. And it was the first kind of biological treatment for cancer back in the 80s for hairy cell leukemia. And people were getting treated with this particular type of immune therapy, before we really actually understood how it worked. And the same has kind of been true for other forms of therapy that are quite common, like chemotherapy and radiation. These were all empirically derived treatments where, you take a bunch of drugs that you don't know what they sort of do, you put them on cancer cells, and you see, the cells slow down their growth or they die. And then, so we know that these are potentially anti-cancer therapies, and we then figure out exactly how they work. So I work a lot on conventional therapies. So understanding particular types of chemotherapy and how they work with an aim to understanding, well, when they don't work, why is that? So why do cancer cells respond to them in the first place, and how do they become resistant to treatment? And I do the same thing with, we do a lot of radiobiology studies. So looking at radiation therapy. So more than 50% of all cancer patients of every kind will get radiation as part of their treatment, and it's a very, very successful treatment for a lot of patients, for a huge numbers, millions and millions of people. But when it doesn't work, we try and figure out exactly why that is. For my research group, we've started to kind of delve down at the molecular level to why that is. So back in the early 2000s, microRNAs were a kind of a class of what we call genetic regulator that were discovered. And, they'd sort of been kind of put on the back burner for a little bit when they were first discovered, because they were thought to be an oddity of some sort of biology. But we now know that they're a cornerstone of normal cell biology and how cells regulate themselves. So back in 2008, 2009, we started working on microRNAs as a major topic in our field, because right around that time, it was discovered that microRNAs were both disruption to microRNAs in cells, in normal cells, promoted cancer and helped cancer develop. So they were causative agents in the development of cancer, but their disruption was also a consequence of cancer. So when cancers developed for other reasons, microRNAs tended to get dysregulated in cells. And one of the interesting things about microRNAs is that while a single microRNA can regulate hundreds of genes, so if you think about the entire human genome is composed of about 3 billion bases. MicroRNAs are only 20 bases. So, you can count them on your fingers and toes. And they're very, very tiny. And yet disruption to one of them, if you get a mutation in a microRNA, it can have a profound effect on the biology of the cell, because it controls hundreds and hundreds of genes, sometimes thousands of genes. So over the years, we've identified several microRNAs in cancer cells that aid in controlling how cells respond to things like chemotherapy and radiation. And one of the other interesting aspects about microRNAs is that you can very easily make synthetic versions of them. So if a cancer cell loses a particular microRNA, which makes it then resistant to, say, radiation therapy, we can make synthetic versions of these and reinsert them, re-engineer a cancer cell to have that microRNA again, a synthetic version of it, and it resensitizes the cell to something like radiation. And we can do this for certain chemotherapies and that. But
how do microRNAs work normally?
So with a microRNA, cells are, the functional aspects of cells and how they operate and how they actually do a job is carried out by proteins. And I suppose the instructions for building that protein comes from the DNA. But there's a type of genetic material called RNA that's kind of an intermediate between the DNA and the protein. So imagine if you think about the DNA like a library, and if you want to make a particular recipe, you will go to the library, you find the right book, you find the recipe in that book. The recipe would be represented by the microRNA. So the library would be, and all of the books in it would be the DNA. The specific recipe you're looking for that tells you how to make a particular, say, meal, is the microRNA. And then obviously the protein would be the meal that gets made. So microRNAs are regulators of that intermediate. So they're a subclass of RNA. So you've got your DNA, your RNA, and your protein. The RNA itself is the message, and that gets translated into the functional protein. But cells have to control that message as well, and how much gets translated into protein. And they do that through microRNAs. So the microRNA will be produced by the cell very, very quickly. It can ramp up massive amounts of microRNA, and that microRNA physically will interact with the messenger RNA that's used to be translated into protein, and it controls how much of that message gets translated to protein. So it can do that by just physically binding to it and stopping the translational machinery in the cell from making the protein.
What else does it do? Can it make other proteins, or just slow down the production or increase the production, or can it make...
It can increase the production. It can slow the production. It can completely stop the production. It can degrade the message or just kind of sequester it away from translational machinery. So we're still, this, it's a 20-year-old kind of field, but in molecular biology terms, it's very, very young. We're still figuring out exactly how they work, and every year we're revising how they work and that. So working in this area can be quite challenging because you're working with the pieces of a jigsaw and you only have certain jigsaw pieces, and every year they give you more pieces and you're like, okay, we're figuring it out as we go. But the biggest part of our work for the last few years has focused on these microRNAs. And even I have a PhD student, I have a couple of PhD students at the moment, and one of the PhD students, David Hackett, David is working on understanding how this particular microRNA that we're focused on, MIR31, how it controls the movement of chemotherapies from the edge of the cell all the way into the nucleus, the kind of the brain center of the cell, to get the chemotherapy into the DNA, where it does the damage to basically destroy the cancer cell. So, David has been working on this now for 3 years, and we've determined, an incredible amount of very interesting information on how these particular RNA controls that.
How do our microRNAs go wrong when there's cancer? What happens to them? What are some things that happen to them?
So microRNAs can be dysregulated in a number of different ways. Quite surprisingly, when you look at where microRNAs are encoded within the human genome, they are their encoding is overrepresented at locations called fragile sites in the genome. So we know in the human genome that there are just over 100 spots that are what we call fragile sites. And those are sites that are more likely to become damaged than other parts of the genome. And these microRNAs, they are kind of also overrepresented in where they're positioned in the human genome. So they do tend to become quite early in the cancer process dysregulated. So that can be through getting deleted. If there's damage and you get deletion of part of the genome, you can lose a microRNA, or they can become mutated, due to things like radiation or chemicals. We can also have them disrupted. So microRNAs and their production in the cell, they have to be produced and kind of from a precursor molecule and matured in order to be able to function. And they get incorporated into little kind of micro machines in the cell in order for them to be able to do their job. So if you get disruption or deletions to parts of the machine that help them do their job, that can also inhibit their function. So there's probably at least 10 different ways that microRNAs can be disrupted. And a lot of the time it's through either deletions or mutations to the areas that encode them within the DNA, but also disruption to the machinery that they need in order to carry out their function.
Well, how does the body attempt to correct this? Does it sense that something's wrong, and does it cause like apoptosis in the cell, or how does that body work to correct this function?
So we do know, depending on the microRNA, so there are thousands of microRNAs now. Most of the time, if you think about it, our bodies will produce over 100 million new cells every single day, and of all of those new cells, something can go wrong in them. And, but the production of new cells and the production of the genetic material, it's very tightly regulated and monitored by systems inside the cell that make sure that it's proceeding correctly. So a lot of the time, if there's a mistake made, say, deletion from the microRNA or a mutation, that gets corrected in the cell. Or if it can't be corrected, the cell will self-destruct. It will undergo apoptosis, as you mentioned.
How fast is the error correction versus the corruption, the flux of those two factors or forces?
I suppose if you think about it this way, so if you take a single cell from your body, every single day there's between 10,000 and 100,000 incidences of damage that's accounted for by, say, mutations to the DNA, which can disrupt a microRNA. And most of those are either corrected or the cell will die. So there's a lot of mistakes that are made, but the fail-safe systems are incredibly efficient. We tend to get, at least a good 40, 50 years before, we really start to see cancer developing in patients. So, the vast majority, we're billions, we're trillions of cells big, and, there's a lot of damage that goes on every single day in all of these cells to our genetic information. And most of that doesn't cause a problem until, kind of much, much later in life.
Would this happen only in liver cells, or is it systematically happening in the body everywhere? Maybe just to a different or greater extent in the liver cells?
Yeah, no, it happens all over the body, to kind of every single cell.
Well, why would some cells be cancerous and some be considered not then?
Well, I suppose we know that, so if you take tumor cells, a biopsy of some tumor cells, and you take a biopsy of the normal tissue very close by, and you look at the mutational burden in the tumor versus the normal tissue, you would probably expect that you would see more mutations in the tumor tissue, but we don't see that. The normal tissue will actually have as many, sometimes more mutations than the cancerous tissue itself. And so not all mutations, I suppose, considered equal.
It is a different mutational profile, but let's say
you have driver mutations and you would have passenger mutations. So driver mutations would be mutations that happen in areas of the genome where it's cancer promoting. So important genes that regulate, the cycling of cells or the proliferation of cells. And then you would have passenger mutations that happen in areas of the genome where there's a lot of redundancy or it isn't really consequential in terms of the development of cancer.
Right, but you'd think the patterning of the mutations in the affected tissue, the tissue that's considered cancerous, would be different from the patterning of the remaining tissue. Let's say liver cancer, the whole liver is not stuck in part of it.
Patterning is different, and that patterning is how we also determine what are mutations that drive the cancer versus mutations that don't drive the cancer. But even within, say, solid tumors, as a tumor grows, it evolves. If you think about cancer cells represented by as people, an individual person, within a tumor, you have multiple neighborhoods where you will have populations, subpopulations, neighborhoods of cells that may be, proliferating and doubling faster than other groups of cancer cells. You will have populations of cancer cells within the tumor that are more resistant to treatment, to a certain treatment, and less resistant to another treatment, which is why we tend to treat patients with, a number of different types of chemotherapy. So because we know that certain tumor cells or certain cells within the tumor will be more responsive and some will be resistant to a particular agent, whereas if we treat with a second agent, those cells that are resistant to one agent might be more sensitive to a second agent or a third agent, and that's why we hit patients with radiation also. So patients rarely get one particular chemotherapy. They will usually get a number of different chemotherapies, all of it with a different mechanism of action, and radiation to try and hit every single population with something that they'll be sensitive to.
Okay. So what do you attempt to do with this? Are you able to do a biopsy of a solid tumor, and based on the patterning that you've seen before, how to attack it with various chemotherapy cocktails? Or what are those ones doing for the treatment?
Well, this is the thing. So, I run a translational cancer research lab. So I'm based with the Department of Surgery. And I work on poor prognosis cancers like pancreatic cancer and esophageal cancer. So these would be cancers that have very, very poor survival rates. And so, while we try to understand exactly why these, particular types of tumor are quite resistant to certain standard treatments, we also try to devise these practical ways to modify patients' responses to those. So we, in unpicking the biology, we've got a lot of models now that basically are developing these microRNAs and synthetic microRNAs as drugs. I suppose genetic-based drugs that can be delivered to tumors in patients that can modify their biological response to radiation and existing chemotherapies. So we have about 3 microRNAs now that we know modify tumor sensitivity to particular types of chemotherapy. So platinum-based chemotherapies, which, have been around for decades and work really, really well in huge numbers of cancer patients, as well as anti-metabolite type chemotherapeutics. But the big one that we tend to focus on is radiation. So radiation is so broadly applicable and used for local control of tumors that we're really trying to identify microRNAs that alter the radiation response of tumors. And we have 4 microRNAs now that we know can do that. And I suppose we're following down the kind of the treatment development pipeline that would go for most other drugs. So microRNAs are really important in not just cancer, but other diseases. So infectious agents like hepatitis C and other types of viruses. So they have entered into clinical trials as other types of as a new type of drug in many different cancers, and as I said, other infectious agents. So the big challenge, like most other drugs for RNA-based drugs, will be actually getting them delivered into the tumor. So right now we focus on, we know that these microRNAs, these synthetic microRNAs, can alter tumor sensitivity to chemotherapy and radiation. So one of my PhD students at the minute is working on what are the best delivery vehicles and that to actually get these microRNAs into tumors inside kind of, a living body. So
well, how do they, do they stay within a cell for their entire lifetime, or do they get packaged out as like extracellular vesicles and transit to other cells and go into them?
Yeah, so we know that we do know microRNAs, even in normal cells, they are externalized in extracellular vesicles. They can be taken up by neighboring cells or in different tissues. An interesting aspect of microRNAs is that you can actually look at the microRNA expression profile of 2 different tissues, say like the liver versus the pancreas, and you can identify, is this a liver or is this a pancreas sample just by looking at the expression of microRNAs. So you will have microRNAs that are solely expressed in particular types of tissue. So for example, miR-122 is a microRNA microRNA that's only expressed in the liver. It's not expressed in any other tissue. And I suppose interesting for that particular microRNA, and that microRNA miR-122 is essential in, say, the HCV, so hepatitis C virus, in its ability to replicate. So in clinical trials, miR-122 has been targeted for inhibition to decrease the burden of the viral particles within the liver. So yeah, we know that there's communication between different tissues via microRNAs and extracellular vesicles. And we know in cancerous tissues that you can get an increase in the release or the kick out of microRNAs in extracellular vesicles. And we also know that certain tumors can co-opt extracellular vesicles that contain microRNAs that they normally don't have, take them up and regulate genes important for, their survival in a way that, you wouldn't have ordinarily thought they can do that. So they can basically suck up microRNAs from other tissues and use those microRNAs to regulate genes inside them.
I've interviewed other researchers where they talked about cancer cells have a very different extracellular vesicle profile. So I guess microRNAs are part of that difference.
Yeah. And, I suppose, considering 20 years ago, just over 20 years ago, we didn't even know these molecules kind of existed. Now we know that they're intricately linked to almost every aspect of cell biology that we can think of, and their disruption has a profound effect on cell biology. So for us, when we think about traditional gene therapy, where you take kind of a synthetic version of a gene and you reintroduce it into a cell, and the cell biology changes in response to kind of the introduction of that gene, that's a singular gene that's being modified that has an effect on the biology of that cell. Here we can introduce a single microRNA, just like you would kind of any other gene. We tend to do it in liposomes, or things that mimic extracellular vesicles. So extracellular vesicles would be natural. We can use liposomes or other kind of delivery vehicles that mimic them in a synthetic kind of sense to introduce a microRNA. You're introducing one single type of microRNA in there, but yet you're having an effect on the regulation of hundreds of genes and changing hundreds of genes at the same time. So that has naturally quite a significant impact on the biology of the cells. Now we look at that in terms of, our downstream interest. So for example, if I'm trying to look at making a cancer cell more sensitive to radiation, I can do that with a particular microRNA, but that microRNA won't just do that. It will do other things to the cell, that could be potentially pro-tumor or anti-tumor. So it's a very, very delicate balance in what we do with these microRNAs. As an example, miR-31, when we put miR-31 into cells, it makes them more sensitive to radiation, but it can make them more resistant to certain types of chemotherapy. So they are a little bit of a double-edged sword.
You said there's thousands of them. So how are you supposed to profile and know what's responsible versus background for cancer?
Yeah, so as I mentioned, a single microRNA can regulate hundreds of genes, but a gene, as any single gene, can be regulated by multiple microRNAs. So the level of complexity in terms of regulation at a cellular level and molecular level is very, very complex. Basically, how we figure out how these microRNAs work is that we do, my PhD students would do a lot of gain of function, loss of function studies, where we would basically take our cancer cells and we either increase the levels of a particular microRNA if they're low, and see how does that affect the biology of the cell. So we would have to characterize all sorts of different aspects of the biology of the cell, and then we would take those sort of same cells and we would suppress the microRNA, or by either deleting it out or doing something to suppress the activity of the microRNA, and see, well, how does that affect? And most of the time, if you increase it and you decrease it, you will see these kind of reciprocal effects on particular aspects of the biology. And then we
also, it sounds like there's redundancy too, of multiple, microRNAs can fit or cause certain cellular functions. So what about the percentages of microRNA versus A versus B? Maybe you could modulate that way.
Yeah, I mean, you have, a very limited number really of microRNAs that can regulate 20,000, plus genes in the human genome. So we also, we have identified, for example, say 4 microRNAs that we know control cellular sensitivity to radiation. But each of those microRNAs does that via a different mechanism. So, one of the complications for us is thinking about, well, if we want to modify a patient's tumor to respond better to radiation, which is the best microRNA to do that? Which is the one that's going to have the more specific effect on that and have kind of less off-target effects that, might be unexpected things that we don't want happening.
But also, too, like, why not do all 4? A little bit of each, or do you have to do a predominance of one? I guess you could do.
We modify these microRNAs, and in trying to understand how they work, we sort of modify them singly, and we look at, okay, well, when we modify one, what does it do to the biology? But we figure out, okay, well, how does it affect the biology such that we can say, okay, well, these 2, say, microRNAs, they work quite similarly in what they do. So there's no point in combining those. But if we have, say, 2 microRNAs that sensitize to radiation, but do so through very different mechanisms, there's a natural benefit there to combining them to see if we can further kind of increase or kind of make the cells less likely to be able to become resistant, say, to a microRNA-based treatment. And again, you are perfectly correct in saying that, like in us identifying, particular microRNAs that alter radiation sensitivity, we do that in patient biopsies and in cell models that we've created in the lab. And while we test them and their function on an individual basis, the way that they work is not, the models that they come from, the biopsies that they come from, the tissues that we've identified these changes in, all of those modifications are happening at the one time. So it may be the case that, you can never truly understand the function of a particular microRNA without modifying, this microRNA and that microRNA at the same time. So it's a very complex area to work in. And I'll probably spend the next 20 years just trying to understand and just scrape the surface of how microRNAs actually alter cancer treatment sensitivity.
What are you using to model this? Are you just culturing cancerous and healthy cells? Are you using organoids of both? Or what?
We tend to start off with patient tumors and biopsies, and we profile and identify a lot of microRNA candidates, so we would take diagnostic biopsies from patients. So these would be biopsies taken when the patients would first come in and they're diagnosed with cancer. And then we follow those patients out and ultimately determine, so we would put their biopsies in the freezer. We follow the patients out and we determine whether they had a good response to treatment or a poor response to treatment. And then we're able to go back to the freezer and separate our biopsies into these are tumors that ultimately are going to respond very well to treatment, and these are tumors that are going to respond poorly. And then we would profile the microRNA differences between those. And some of them will just be kind of passenger, kind of changes that, come up as statistically significantly different, but they don't have a particular bearing on treatment sensitivity. But then others we would basically test in simple models, say cell lines, in 2D culture, and then we would move into kind of organoids, and then we would go into kind of animal models, ultimately, going back to kind of humans. It's not always straightforward. So for example, MIR330 is a microRNA that we found to be deleted in 100% of patients with esophageal cancer who don't have a good response to chemotherapy and radiation. So we modified, we genetically manipulated that particular microRNA in 2D culture and looked at sensitivity to treatment, so all sorts of chemotherapies and radiation, and basically it seemed to do nothing, which was very kind of surprising to us. We knew it was actually working, when we would manipulate it, we saw changes to biology, but ultimately the the cells weren't any, more or less sensitive to chemotherapy or radiation. However, when we actually grew those tumor cells in a living organism, so in a mouse, and created 3D tumor microenvironments, that's where we saw the difference, the biological difference, and the assertion of the effect of the microRNA manipulation. And so the 2D culture, wasn't an appropriate model there. When we actually went into, growing a tumor with the vasculature, with hypoxic areas, with a metabolic profile and an acidic microenvironment, that's where we found now actually MIR330 has an effect on treatment sensitivity to, say, chemotherapy and radiation. So our choice of model is very important and can dictate essentially what we see. So we tend to have to use cell lines, organoids, and animal models, whether it's zebrafish or mice, in order to understand the effects of a microRNA on biology.
Are you looking at all the microbiome? Each cell would have pretty much its own. I know from talking to some scientists that tumors have their own microbiome that's distinct from healthy tissue surrounding them.
Yeah, well, we work, my group, we work in GI cancers, so gastrointestinal cancers, and so if you look at the lower GI, you have an entire ecosystem of at the microbiome level, and the same would be, for the esophagus, the food pipe connecting the mouth to the stomach. My group, we don't look at the microbiome. Is this important? Absolutely.
Okay, there's just only so much you can do, so you got to focus.
Yeah, that's it. Yeah. The years go by very, very quickly,
Okay, well, very good. Understood. What's the best way for people to learn more about your research and keep tabs on it?
So I suppose if you want to learn more about my research, I have a Twitter account, SG Maher, and I also have a LinkedIn profile, but you can go essentially to PubMed, type in Maher SG, and that will take you to kind of all of our research.
Okay, very good. Well, Stephen, thanks for coming on and explaining this complex topic. I appreciate it.
Thanks for having me, Richard.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.